Beardsley coming at you live from Mutation Station. For this week's episode, we will be talking about small-scale mutations. These specific mutations include point mutations, or more specifically, changes in the single nucleotide pairs of genes. And these point mutations themselves can happen in two different ways, via base pair substitutions or base pair insertions or deletions. One nucleotide base is altered in base pair substitutions, resulting in either silent, missense, or nonsense mutations on the amino acid level. In silent mutations, the alterations in structure of these proteins goes unnoticed when observing the function because of the fact that there can be many codons for one protein. For example, there are 6 different codons that code for leucine, and so if one of these codons is altered to resemble the other, the protein will still be coded for. In missense mutations, one amino acid is altered. As a result of this change in structure, there's a change in function that may lead to issues later on. In nonsense mutations, a stop codon halts protein synthesis, resulting in an incomplete protein, unable to function. Point mutations are also manifest in base pair insertions and deletions. In insertion, a nucleotide is inserted into the DNA, and in deletion, a nucleotide is deleted from the sequence, both resulting in what is called a frame shift mutation. Frame shift mutations occur because of the frame of the DNA sequence. It's literally shifted to the left or to the right. This addition or subtraction of a singular nucleotide messes up the whole DNA sequence. These frame shifts, similar to the substitutions, can result in missense and nonsense mutations in amino acids as well. It's also important to note, before we move on, that these minute changes in DNA strands can have detrimental effects on any organism, like how sickle cell anemia, for example. It really just boils down to the DNA giving faulty instructions to the mRNA, forming a misshaped hemoglobin protein.
Greetings, this is Caroline Brogden reporting live from an animal cell. Let me tell you, we've got some wild activity out here.
Let's take a look at mitosis, the process by which somatic cells reproduce. In the cell cycle, about 90% of the cell's life is spent in interphase, a process divided into 3 distinct steps called G1, S, and G2. G1, gap1, you've got replication of the cell's organelles and cytoplasm. S, synthesis, the DNA of the cell in the form of chromosomes replicates. G2, gap2, the cell makes proteins, continues to grow, and prepares for division. Then it's the mitotic stage, which includes mitosis and cytokinesis, and the cell divides into 2 diploid daughter cells, meaning each is a complete set of DNA. In humans, that's 46 chromosomes. The cell has thus completed a full turn of the cell cycle, and between the phases are checkpoints. The cell will become arrested and stop moving through the cell cycle if, for example, there's damage to the DNA. This is regulated by, you guessed it, proteins, specifically proteins called cyclins and cyclin-dependent kinases. Backing up for a moment, we learned that genes code for amino acid production, which form proteins. Your DNA contains genes called proto-oncogenes, which are responsible for normal cell growth and division. Transformation, or conversion of proto-oncogenes into cancer-causing oncogenes, will abnormally stimulate the cell cycle. It affects coding for amino acids, which affects the type and amount of proteins produced, thus altering the cell cycle. Transformation is a result of mutation, like movement of DNA within the genome, or amplification of a proto-oncogene, i.e. increased number of copies of that gene, or even point mutation. So what happens when your proteins are out of whack? The cell cycle doesn't become arrested, and the cell just keeps dividing. In simplified terms, cancer happens. A lot of mutations are necessary for cancer to occur, but at the DNA level, cancerous cells have active oncogenes and mutated tumor suppressor genes. Here's a common example. Mutations in the RAS proto-oncogene lead to hyperactive RAS protein activity, increasing cell division by moving the cell past the checkpoints at an accelerated rate. And P53, a gene that prevents cell division from occurring to avoid passing on damaged DNA, becomes mutated and the cell divides uncontrollably. People can inherit these mutated alleles too. Bottom line, cancerous cells are uninhibited and insufficiently regulated by proteins, so they divide abnormally, and that's not good. Thanks, Cami. Back to you.
Breaking news! In sex cells, similar processes occur. In meiosis, which is the reproduction of sex cells, the number of sets of chromosomes reduces from 2 to 1 in the gametes in order to counter the doubling that occurs in fertilization. But when the members of a pair of homologous chromosomes don't come apart properly in meiosis 1, non-disjunction occurs, and from this, a plethora of things can go awry. The gametes will either end up with 2 of the same type of chromosome or neither, and this results in a condition we call aneuploidy, when a zygote has an abnormal amount of chromosomes. When 2 gametes come together and one is missing a particular chromosome, the resulting zygote is considered monosomic for that chromosome. Similarly, when 2 gametes come together and one has 2 copies of a particular chromosome, a trisomy occurs. Polyploidy is a result of complete non-disjunction in both gametes. It's when a zygote has two complete sets of chromosomes in its somatic cells, like triploidy and tetraploidy. This non-disjunction can result in various conditions, such as Down syndrome, which is a triploidy of chromosome 23.
We interrupt this program to inform you of a mutation that has occurred in Mutation Station. Hope you caught the deletion. Signing off from 101.1.